Good afternoon. Beginning in 2018, the Purdue Engineering Distinguished Lecture Series invites world-renowned faculty and professionals to Purdue Engineering to encourage thought-provoking conversations and ideas with faculty and students regarding the grand challenges and opportunities in their fields. Besides presenting a lecture to a broad audience of faculty, graduate, and undergraduate students, they engage in an interactive panel with faculty and students. It is my pleasure to introduce Dave Barr, Senior Associate Dean, College of Engineering, and Professor of Materials Engineering.
Thank you, Kevin. Really appreciate it. It's my real pleasure to introduce today's speaker for the Purdue Engineering Distinguished Lecture. We have the privilege this afternoon to have Dr. Christine Schmidt. Dr. Schmidt is a distinguished professor in the Creighton Pruitt Family Endowed Chair and the former department head at the University of Florida for biomedical engineering. Prior to joining the University of Florida, she was the Goodrich Endowed Professor at the University of Texas, Austin, in both biomedical engineering, she was a founding member of that department, and in chemical engineering. She holds a PhD in chemical engineering from the University of Illinois Urbana-Champaign, so she's familiar with the land of cornfields and Midwest activity, and her BS in chemical engineering is from the University of Texas. It is rare that we get to introduce somebody who's a member of both the National Academy of Engineering and the National Academy of Medicine, but today we have that privilege. She's a fellow of more societies. If I list all the societies, her talk will get shorter, so I'm just going to go ahead and point out from AAAS to AIMBE to BMES. She's very well respected within the field. She's been very active within the state of Florida and is also on the Florida Academy of Sciences and provides expertise to serve the entire state, not just the university. Her research is focused on biomaterials for neural tissue, as we'll hear, and her work is the foundation of products that are in the market, the Avance Nerve Graft from Axogen, and the VersaWrap protector from a startup company, Alafair Biosciences. So with that, it's my great pleasure to introduce Dr. Schmidt and welcome her to today's lecture.
Thank you. Thank you so much. Hello. Can you guys hear me? All right. You're doing the magic over there. Thank you. Okay. So thank you for that introduction, and thank you for the invitation to be here. It's a real honor to be here. And back in the cornfields, as you say. And in the perfect time for corn mazes. You guys should take me on that tonight. No, okay. So what I want to talk about today is some of our research and the pathway that we have taken over the years to get some of this research into the clinic, which has been a huge passion of mine and a large focus for biomedical engineering in general. So the space that we work in is called neural tissue engineering. And so we work in both peripheral nerve repair and trying to find ways to encourage peripheral nerves to regenerate, and we also work in the area of spinal cord injury repair. So I'll talk a little bit about both of these. The first area I want to talk about is in the area of peripheral nerve regeneration. And so to give you some background and context to this, peripheral nerve injury, there's actually quite a few peripheral nerve injuries that happen every year. And these can come from a multitude of different types of scenarios ranging from cancer to traumatic injuries, congenital defects where somebody needs to repair a nerve in the body and to regain function. So there's a number of injuries. Peripheral nerves have a little bit of ability to regenerate on their own, but not completely, especially if it's a complete sever of that nerve or a more traumatic injury. So what happens is after the peripheral nerve is injured, the axons that are part of the nerve die off downstream of the injury site. And so what we're trying to do is find ways that we can bridge the gap, the injury gap, and allow those axons to grow down the injury back to the muscle target where they have lost that contact. So the whole goal is to provide a bridge and to allow for this regeneration back to the distal target so that the patient can regain function. So when we started this work many years ago, the traditional approaches to repairing a damaged peripheral nerve consisted of either autologous nerve grafts, where they take a piece of peripheral nerve from somewhere else in your body that you may not miss, like a small peripheral nerve in your leg that controls sensory function in your foot, and then transplant that to the site where you have the injury to regain motor function of your hand, which is very important. There were also a few artificial conduits that were available, and these consisted of mainly what we call just hollow conduits, whether they be collagen-based or a synthetic material. And those, again, serve the function of providing that bridging the gap between the two damaged nerve ends and providing a pathway or an internal sidewalk for those axons to regenerate so they can get back to the muscle target. So, and there's pros and cons to these. The peripheral nerve, the autologous nerve graft, I should say, obviously does very well, and that is the clinical gold, had been the clinical gold standard at the time, but requires 2 surgeries, so you're losing some function from somewhere. It may not be major, but you're still losing function. And then there's the possibility of, infection at that particular site as well, and it's also more time-consuming and costly. And the artificial conduits, although they do well, they only work over certain distances. So it turns out that the artificial conduits only work over very short defect sites, so they're not applicable in a lot of cases where you may have to use the autologous nerve graft. So there was a gap here. There was a need for something that was different and could provide options for patients that these particular options did not provide. So to give you some context as to why those hollow conduits don't work that well, so regeneration in a hollow conduit, what happens is if you suture the hollow conduit across to the 2 damaged nerve ends, what happens is that the conduit fills with blood and proteins from the surrounding tissue, and you get fibrin, which is naturally in blood, coalescing into a blood clot, essentially. And this blood clot forms this fibrin cable between the 2 damaged nerve ends, and then that fibrin cable serves as the pathway to allow for regeneration of the cells and the axons that are in the peripheral nerve, the surrounding peripheral nerve. So you get migration of endothelial cells and Schwann cells and fibroblasts, but then your axons are regenerating along that pathway. So cells need something to grow on, and that fibrin cable provides that pathway. But unfortunately, that fibrin cable, as the defect is larger and larger, doesn't form. So over very large defects, you don't get a fibrin cable, and if you don't have a fibrin cable, there is no scaffold for the regenerating cells and axons, so you don't get regeneration. So what this means is that a fibrin cable is essential for regeneration because it provides that pathway, and it can't form over longer distances. So what we decide, or so what is necessary, is that you need some sort of internal matrix to allow for this regeneration. And that's why these hollow conduits are limited to very small defect sizes, okay? So what we asked ourselves is, can we do better? Can we do better than the autologous nerve graft? Can we do better than these hollow conduits? And so we took out an approach that was basically 2 parallel approaches to solving this problem. One was the top-down approach, is can we take native nerve tissue and remove the parts that cause an immune response so that you have something off the shelf? So we know that the autologous nerve graft works very well, and it was the gold standard because it has an internal matrix, right? The only thing is you don't want to take tissue from the patient. And so could we have something off the shelf, maybe using cadaver tissue, that would work as well or almost as well as the autologous nerve graft? And so that was coming up with this decellularization approach. Our bottom-up approach was, can we use the materials that we work with in the lab and engineer a matrix that has this internal structure that is lacking in the hollow conduits and engineer another type of guidance channel that can be used in the clinic. So with our top-down approach, as I mentioned, autologous nerve tissue is the gold standard. So can we take that, strip it down to its basic skeleton, which is the extracellular matrix proteins, the collagens, the laminins, leave behind that structural support that provides the guidance, but remove the cell components that cause the immune response. And the goal was to preserve this natural microarchitecture as much as possible. And I'll show you in a minute why that's important, because nerve has a lot of microarchitecture that's critical for its function as well as for its regeneration. So we'd started this project. At the time, decellularization hadn't been done on a lot of tissues, so especially larger three-dimensional nerve tissues. There were skin products and thinner types of extracellular extracellular matrix-based products, but not really larger 3D tissues. So what we did is I had a fantastic chemical engineering graduate student who worked on this and iterated on how can we remove the cells but not damage the intricate microarchitecture of the nerve. So you'll see in these pictures, so on the left in the fluorescence image is the partial cross-section of a peripheral nerve. Okay, similar to the peripheral nerves that you have in your body. And the red is showing the neurofilament, which is the interior cytoskeletal components of the axons, and the green is showing S100, which is basically a Schwann cell marker. So these are the cellular components of the nerve that we want to remove. Those cause the immune response. On the right side is the staining of just the collagen. And so that's the intricate microarchitecture we want to preserve so that we can have these pathways for the axons to regenerate. So you'll see that there's a lot of organization here. This is much different than trying to decellularize skin, which is very amorphous and doesn't have a lot of architecture. So it took a while to come up with a formulation where we could remove the cells but not destroy the microarchitecture. And so we can effectively remove the cells and then keep that structural preservation, so teasing out those cells, in a sense, very gently, so that we can have that scaffold for regeneration. And the way we did that was having different types of detergents that have multiple different charges so that we can have tight, small micelles that can penetrate this dense nerve tissue. Nerve is also very lipid-rich, unlike something like dermis. And so being able to get the detergents in there effectively and then rinse them out without destroying this microarchitecture was critical. So, and this just shows in more detail how we were able to do that. You see native nerve, that's a cross-section of native nerve. Here we're staining for basal lamina, which is the individual laminin coating around each axons. And so that's a structure we want to preserve. And then with our optimized technique of decellularizing, we can retain that spherical microarchitecture. It's a little bit different because we've decellularized it. But then you can see that other decellularization methods that people had attempted with nerve and other types of tissues was leading to decellularization, but leaving behind a lot of microfracture of the actual ECM, the extracellular matrix. So our hypothesis is that we wanted that extracellular matrix because we want the axons to proceed on a pathway, a continuous sidewalk versus a sidewalk after an earthquake. So. And then we were able to show that we were able to regenerate using this, so we could take these decellularized tissues and then implant them into animal models where we sever the sciatic nerve, in this case, in rats, and then show that we can have regeneration in our optimized, our decell tissue that's comparable to what we would see with an autologous nerve graft. And again, that's the gold standard in the clinic. So we're able to do that. And what we also showed is that other techniques, decellular techniques, such as freeze-thaw and other chemical decellularization techniques that led to this microfracture that I just showed you, they retain the components of the ECM, but not the microarchitecture. So it's not just the composition, it's the actual, again, the architecture there. So we didn't have as much regeneration in those. So what this was telling us is that that intact microarchitecture is important. Not just having the ECM there, but having it in an organization that the axons can regenerate. And so at this point, we did our due diligence and we filed our intellectual property. We always do that. So we did our disclosure, and then we published our paper. And a company out in Florida, so we were doing this work in Texas at the University of Texas. Then a company based in Florida, Axogen, called us up and they said, "Do you have this? We just read your paper. Is it protected?" And we're like, "Yes." And they're like, "Great, we're going to be there." So they basically came and visited our labs and said, "We want to license this under the condition that we can get this to work with human tissue," because everything we had done was with rat tissues. We're an academic lab. So we said, sure, let's do this. And so they basically converted our lab into like a GLP type lab where they worked side by side with my students. We had separate cabinets that we stored their chemicals in, and everything was separate. And did this on human tissue. And that was like very exhilarating, but also like, what if this doesn't work? Because we found out that human tissue's frozen. They have to test it for pathogens. They freeze it. We didn't do that. We just pulled it out of the rat and decelled it. So and also the human tissue is bigger. We're like, oh my gosh, it's so much larger. Is it going to effectively penetrate the tissue, the detergents? So fortunately, we were able to show that we could decellularize human tissue and that we didn't get a lot of, we were able to get rid of the immunogenic cell components, leave behind the microarchitecture. The freezing didn't seem to have an impact. The size we were able to scale it up. And so at that point, it just went to Axogen. Okay, they kept us in the loop, but they licensed it and they said, okay, now we're going to take it and make this into a product. And so again, we published our results in 2004. They came and approached us, shortly after that. And then it was first in patient in 2007. And that was scary because they called us up and said, it's going into a patient, it's going in Dallas. And I'm like, oh my God, I need to go there, but I didn't go there. And I was like, oh my gosh, who is this saving? Is this a young child who's had some sort of major injury or somebody who's had, cancer surgery? And they're like, well, it's somebody who was in a bar fight. And I'm like, do we really? No, okay. So I said, great, however it works. So that was exciting to have it first in patient. And I won't tell the story now, but it went into the patient very quickly because it was at the time regulated as a modified tissue transplant. And then since then it has undergone multiple regulatory changes in the process, but it got it into humans first, fast, and that was fantastic. And you'll see from the picture that it feels and like native nerve, and so the surgeons like working with it better because they're used to working with, graft material, autologous tissue. So, and so here I just want to say it's about the impact on the patients. These are a couple of applications that have come since the bar fight. So maybe a little bit more, you feel a little bit better about this. A young child, 12 years old, in an ATV rollover accident. That could have been my kids, so I'm like, because my husband does crazy things when I leave. So needing, basically it had a 7 centimeter defect in his ulnar nerve in his arm, and it's been used in that case. And then this is another very common example is the military. So this is used a lot in military when they have blasts or other types of injuries, gunshot wounds. And here he was facing possible amputation of his leg, and this was able to save his leg and allow him to walk again. So that's what we're doing it for. But a couple of side notes before I switch to some of the other things that we've done. At the time we were doing this, decellularization seemed obvious, okay? There was other decelled tissues out there, and some people had published on decelled nerve, but it was those microfractured tissue, and it wasn't in the clinic. So it's like, people are like, this is so boring, you shouldn't be working on this. It seemed like, it must have been done. But when you dig deep, there was nothing in the clinic. And we said, no, it hasn't been done, and no, it may not be as sexy as some of the other work that we were doing in the lab. We were doing some work with quantum dots and all sorts of cool things, but this could have a clinical impact. So again, like we were told, it's not sexy. This was pre-tenure that I was working on this. I had warning from senior faculty, you should not be doing this. Don't do this. This is going to kill your tenure case. We never got funding directly for this work. I siphoned money off of NSF. They were okay with that. So, and our earlier manuscripts don't have as many citations as some of the other, what I would call sexy work that we've done in our lab. And it's good to have multitude of different projects, and it's good to have near-term and long-term. And I always call it my balanced portfolio. But it's also good to pursue what you feel is important for you as well. So the bottom line is the obvious may not be done. Simplicity and unexciting can have their rewards. And that funding and citations don't correlate to impact as much as, in my mind, as the impact on the patients. So that was a fun thing. But at the same time, we were doing a lot of other really exciting projects in our lab. And we had other approaches that we're looking at for peripheral nerve repair and other types of wound healing. And so we thought about longer defects. So the Avance graft from Axogen, the decell nerve product, works in much larger grafts or much longer defects than you would see as one of those hollow conduits, okay? 7 centimeters, sometimes 10 centimeters, but it can't go to like 30 centimeters. And that's because it's decellularized, it doesn't have cell components and growth factors. So we were saying we need to look at even larger defects. There's a smaller population, but it's still an important population for those patients. And so there we were looking at our bottom-up approach. And here what we're trying to do is say we have a lot of cool materials in the lab that we work with that we could build something and engineer something that looks and feels like nerve. And if we do it from the bottom up, we can incorporate growth factors, we can incorporate cells, and hopefully we can have regeneration then over longer distances. So that's been our goal, and we've worked with hyaluronic acid for a lot of this work, and most of you are probably familiar with that, but it's basically a ubiquitous component in your extracellular matrix. It's a sugar molecule, and it's essentially non-immunogenic because it's a sugar molecule and not a protein. And it plays already a vital role in wound healing in your body. It plays a vital role in embryogenesis, so it's known for helping under those conditions where you're trying to regenerate. It's also, you can scale it up pretty easily, so that's good from a production standpoint. It was already FDA approved in other indications, so that makes it a little easier to get into the clinic. And other things, it's non-adhesive essentially, so we can then program in the types of adhesion we want that gives us low background in a sense. And then so that we can combine it with other materials to make it permissive for peripheral nerve regeneration. And it's degradable, so ultimately we want something that degrades, put something in the body, help the body heal itself, and then disappear over time. So we've worked with hyaluronic acid in its cross-linked form. So hyaluronic acid in your body on its own is very labile, meaning it degrades very rapidly and turns over. So we work with it in cross-linked form so that we can stabilize it for implantable materials. And so we do a number of different ways of cross-linking it. One of the ways is using glycidyl methacrylate. We add that moiety to the backbone of our hyaluronic acid, and that gives us what we call GMHA, or glycidyl methacrylated HA. And then we can use that as a precursor and then UV cross-link chains together. So, and then we can control essentially what kind of gels that we get. We can control and make them high swelling, low swelling. You have a lot of cross-links, it's going to be lower swelling than fewer cross-links. So we can control the degradation properties, the swelling properties, and so forth. So swelling, degradation, and mechanical properties. So it makes it tunable and something that's going to last longer. And as you know already, I've told you that nerve has microarchitecture. So these are pictures of the basal lamina tubes. The red is showing the cross-section staining, and the bottom is an SEM showing the longitudinal. So lots of architecture. So one of the things that we've really done is when we're trying to build up from the bottom is make sure we incorporate that microarchitecture into our structures to replicate that nerve structure and allow for the regeneration of the axons. And so we've done this in a number of different ways. How can we incorporate this microarchitecture? We've used things from multi-photon fabrication, in situ crystallization, magnetic alignment. I'm only going to talk about the last two. I'll tell you a little bit about in situ crystallization, just touch on magnetic alignment and how we're trying to incorporate this microarchitecture. So with in situ crystallization, I have a very talented, also a chemical engineering graduate student, who was trying to impart microarchitecture in different ways into our hydrogels, because otherwise they're just amorphous. And if they're amorphous, they don't provide that directionality for our axons. How can we do that? And so he came up with a way of incorporating crystallizable molecules, such as urea, that we could put into our precursor hydrogels, and then essentially crystallize those molecules inside our hydrogels, then cross-link around the crystallized solution, and then rinse out the crystals to leave behind a hydrogel that had the imprint of the crystals inside them, or the network of crystals inside them. And those would be pathways. Our goal would be that those would be pathways for the cells to regenerate along. And so the nice thing is it's simple and it's inexpensive. It doesn't require a lot of equipment, no organic solvents. That's good if you're trying to move things into the clinic. And again, so basically rinsing these out and leaving behind a hydrogel with micro patterns. And so we were able to show that we could create different microarchitectures depending on the concentration. If we have low urea concentration, in this case, we would have more arbor-like structures. And then if we increase the concentration, we get really tight structures that are parallel, which might better mimic, growth paths for axons in a long conduit that we want to create. So this is great. And we were able to show that we can get cells to grow on them and follow these patterns. So this is just thin films of our patterned gels showing that we can get fibroblasts to grow on them in the direction of the alignment that we have created. And otherwise, the cells would be all over the place and not have any directionality. So we're like, this is great. We can control cells, all these wonderful things, but we've got to make it bigger. So at the time, we were making thin hydrogel films. We said, oh, we need to make these larger for nerve. Nerve is, it's not this big, but, it's still three-dimensional, okay? And so we had to take it to larger structures. And then we went to trying to create this alignment in these larger structures, and these are the types of crystals that we were getting. We were having a very hard time when we add that third dimension of controlling the crystal microarchitecture. So it basically presented challenges doing this. And we said, well, this isn't really working out well for nerve. But what we found is that the films, the thinner films that we were working with, had some really interesting properties. We're like, that might be interesting in and of itself. So even though it's not ideal for nerve, it's okay to get sidetracked. Okay? So my whole life is about getting sidetracked. But and so like I said, we found out that these films had interesting properties. They were pliable. We could put a suture through them. They were strong, and we can, the incorporation of these microchannels were helping there. We also looked at adding alginate to this and did some really cool things and said, well, okay, it's not working for peripheral nerve, but these films have to be useful for something. Because keep in mind, it's based on non-adhesive HA, hyaluronic acid, so it's non-adhesive. So we found out that these films, again, flexible, robust, elastic, and suturable. And so we just essentially went to our local hospital. We didn't have a med school in Austin at the time. We just went to the local hospital. Of course, we went to, a neurosurgeon because we're neuro people. And we took some of our samples there and we said, is this useful in any way? What could you use this for? And so he says, well, I do these back surgeries, right? And I do brain surgeries. And what happens is after the surgery, I get a lot of adhesions forming. This is basically scar tissue that's post-operation. And I have to sometimes go in there and do the surgery again. And that's not a good thing. No, you don't want that surgery again. And he said, the product that's out there is terrible. He says, I hate it, I hate it, I hate it. It just crumbles up. It's not, I'd rather not use it. So he said, this might be useful. And so he essentially, introduced us to this whole idea of could we block scar tissue? And we hadn't thought of that. We're thinking encourage nerves to grow, axons to grow, not blocking tissues from growing. So he literally came into our lab and worked side by side with my students and tested things. He says, no, I would never use this in a clinic. Oh, this feels better. Okay, this is that. And so we iterated with a surgeon in the lab. And then when we did that, we said, okay, now we have to sterilize it. We're like, oh, shoot. So, so then we sent it off for, e-beam sterilization, and the whole mechanical properties changed. So then we had to go back to the drawing board and do it again. But it was great having the surgeon in there. And then he had surgeon friends who then scored blindly all of our animal studies. So that was really cool. And they also have lots of money. Keep that in mind. Okay. Which is good when you're fundraising. So he introduced us to this whole world of adhesions, from the back and the brain, but also it happens in other areas. The abdomen is a huge area, shoulder surgery, knee surgery, tendon surgery. So many areas that adhesions are a huge medical issue. And this is something we had never thought of. And so just that alone was really cool to bring us into a different space. So many years later, I had a fantastic PhD student working with me, Sarah Mays at the time, who was iterating with the surgeon side by side. And we had to keep things separate. We had her Alafair stuff, we had her PhD stuff, but then when she graduated, she became the chief scientific officer at Alafair. And then in conjunction with their CEO and our surgeon friend, we were able to, this company, Alafair, was established that took this product from something in the lab to something that could actually help patients. So, and Sarah's a huge source of the success of this because she's the one. I didn't want to quit my day job. I love being a professor, I did not want to just stop and start a company, but I want to see things get out there. So whether somebody licenses it or somebody else does a startup company, that's great, but I was not going to stop that. And at the time, all of this was happening also at the time that I was moving to Florida to be department chair. So definitely couldn't have that on my, that was just too much to handle. So she wanted to do this, thank goodness. And she has put blood, sweat, and tears into this and has seen this literally go into patients. So this is the product VersaWrap. So it's based on these sheets. It's hyaluronic acid and alginate. So we've iterated a lot on it since the initial days, and it's now approved for use in tendon, ligament, muscle, nerve root in the spine, and so forth. And so it first went into patients in 2017, and it's been implanted in over 35,000 patients today. And they just got approval for VersaCoat. So that's just a recent FDA approval where they can now use this in more in areas where it's harder to get to, where you couldn't maybe put a membrane, but you can squirt some material in there. So it's been highly successful so far, and so we're excited to see that, and I'm so proud of her. If you ever have a chance to invite her for a talk at some point in the BME department, she gives a fantastic talk. Okay, enough plugging her. So I want to go back to my nerve repair quest. So this is something I'm passionate about nerve repair. The adhesion barrier and VersaWrap and Alafair was really cool, but that was kind of that was a side project in a sense. I want to get back to where my roots are in a sense. So this is now at Florida and still pursuing this, and how can we create architecture for longer distances to guide axons. And so here I met up, in the magnetic alignment area, I met up with a colleague, Dr. Carlos Rinaldi in the chemical engineering department. He does work in magnetic nanoparticles. Specifically, he's worked with targeting them for cancer, right? So cancer detection. We're like, oh, that's cool. He and I are just talking, and we're like, can we use your magnetic particles to maybe work as a sacrificial template so that we can create long-range architecture? He said, let's try it. So talk to people, go to seminars. This is all really important. So another just random collision. So we work together where he's the magnetic expert, where we're incorporating his iron oxide nanoparticles into alginate to create larger magnetic particles that are on the same size scale as the types of channels we want to create for axons. And then using magnets to align them. And then once they're aligned, what we do is we use alginase to get rid of to degrade the alginate, and then we rinse out the iron oxide nanoparticles and leave behind just a template of hollow channels that can help guide our axons. So this is a technique that we developed together that it gives us roughly linear architecture. We can scale it up, and it's a way that we can also incorporate biochemical cues within our scaffold so that we can hopefully get longer range regeneration. And so this is what this looks like. We started out with these small magnets. We put our hydrogel in there with the precursor, and then we put it in the magnet, and we get this alignment. And so you'll see right after cross-linking, those are the iron oxide nanoparticles that are black, aligned channels. We can remove those. We have now pores, and then we can backfill the pores to show that, yes, the pores are open. And we tested this, again, in a sciatic nerve injury model. Again, I'm interested in peripheral nerve regeneration, where we stick in one of our, this is a hyaluronic acid-based hydrogel that has the templates in them, and then we also stuck in a hyaluronic acid-based hydrogel that doesn't have the templates, so it doesn't have those channels. And what we saw is that we can get a lot of growth with our magnetically templated, but we don't see the same growth for these channels when it's not magnetically templated. So these pathways are opening up areas for the axons to regenerate, whereas the material itself is not allowing for this regeneration. And then showing that we are actually getting axons and Schwann cells growing through those channels and not just scar tissue, because that's very important. So that was very encouraging. We've been working on ways to better understand this, better control the microarchitecture, or better control the channel alignment in these materials. We've also been looking at how can we scale these up to longer distances. So, Dr. Rinaldi, with his magnetic expertise, is able to create magnetic, what we call film wheels, so that we can get longer range organization and alignment of these channels. So you see on the top our original alignment length is sort of limited to 1 centimeter, but then when we use this film wheel type of magnetic structure, we can get really long alignment of our structure. So we can scale this up to longer distances, hopefully that are more clinically relevant. So that's, where we are there. So we're still working on that. That's active. So just quickly, and then I'll tell you a little bit about the spinal cord, just touch briefly on that. But just, we can do better. So I showed you how we were able to use a top-down approach to develop a decellularized nerve. That's now the Avance Nerve Graft. And then use the bottom-up approach, which we're still working on with magnetic alignment, but then we had a deviation or change of plans where we went and developed an adhesion barrier as well. So licensed to Axogen. We had some failures, but sometimes it's good to turn those failures into some successes. Magnetic templing, we're not sure, and then some other applications, so led to VersaWrap and to VersaCoat. So that was sort of our peripheral nerve story. It's still ongoing, but that's a little snapshot of what we've been doing there. And just very briefly, I want to tell you some of the work we're doing in spinal cord injury, which is our current focus. So here, we are very interested in creating injectable formulations that we can inject either into the spinal cord or just on the outside of the spinal cord in the most minimally invasive way possible, that we can encourage decrease in inflammation. That's a huge barrier for spinal cord injury, but also to promote regeneration and possibly use our injectables as a drug delivery tool. So we have this decell nerve that we love, and studies have shown that peripheral nerve grafts in spinal cord are beneficial. So we want to use that as a starting point. And so we have our decell peripheral nerve that we're actually making into, that we digest it down into formulations that we can make into hydrogels. And so we can make them into in situ injectable hydrogels. And so the whole goal is that we can, again, in a more minimally invasive way, apply these gels that have their own inherent properties. And I won't show all the data, but we've shown that these gels do decrease inflammation and they can promote axonal regeneration, but then also use it as a carrier for either cells or drugs for different types of applications. So you'll see here the nice thing about the injectables is that it can conform to irregular spaces. It's a little bit less invasive. Of course, anything going in the spine is pretty invasive, but less invasive than other types of approaches, and that lowers the risk of potential injury. And so we've worked with Dr. Mary Bunge. She has since passed, but she was at the Miami Project to Cure Paralysis, to show that we could use these injectable materials to transplant Schwann cells. So the Miami Project had a large clinical trial of transplanting Schwann cells into patients, and they were using saline. So saline, injecting Schwann cells, and finding that the Schwann cells, it didn't have as much efficacy as they wanted. And in the studies that they did in rats, they were showing that the cells were just dying. They weren't surviving the injection. And then once they got in there, they were basically just going all over the place and not staying at the site of injury. So I gave a seminar on injectable hydrogels there. So go give seminars, give talks, meet people at conferences. Gave a seminar there, and she said, let's get together. And I said, let's do that. Can we use the injectable hydrogels that we'd already formulated to deliver these Schwann cells? She'd been studying Matrigel to deliver, and she says, oh, the Matrigel's doing such a great job, but it's not clinically translatable. And I said, you're right, it's not. So we worked together to show that with our injectable peripheral nerve, that it protects the cells during the actual injection process. So it keeps them protected, and so they survive the injection process, but also it retains them at the site of injury after a spinal cord injury and promotes regeneration. And so comparable and actually better than Matrigel, which was not clinically translatable. So this just shows this is some of the studies we did to show with spinal cord injured rats that we can get better recovery with some of these injectable peripheral nerve grafts. And so this was very exciting. So now we have moved this to both porcine nerve, scaled it up to porcine nerve, as well as to human nerve. And we're doing a lot of preclinical studies to show that this is looking very exciting. And we're also combining this with anti-inflammatory drugs, as well as with Schwann cell delivery for hopefully improving patient outcome and spinal cord regeneration. So again, the whole goal is to develop a gel that can be used with multiple different things. I mean, on its own, we've shown it's anti-inflammatory and pro-regenerative, but we also want to use it as a delivery vehicle for X. X could be a drug, X could be Schwann cells, X could be, whatever kind of cells you want, and hopefully help spinal cord injury patients. So right now we have a pig study that's going to be starting in February. So look at our cute little pig. What we've learned in this process is pig studies are very expensive. We're getting a discount with Dr. Candace Floyd because she is super kind. She has a fantastic GLP neurotrauma facility at Emory University, and she's giving us a huge discount to do a trial study of 3 pigs that we've gotten some money for. So it's normally $50,000 per animal to do this study, and she's giving it to us for $17,000. It's like BOGO. Okay, so that's scheduled for February, where we're going to be looking just at the material itself. Then the goal is to use, if that looks good, and it may not, sometimes things just fail and don't scale up, and that's okay. But we're going to see. And if it does look good, if the results are encouraging, then the next step is to look at as a delivery vehicle for cell and different types of therapeutics, and possibly partner with others who are developing therapeutics for delivery to the spinal cord as well. So hopefully I gave you a little taste of what we've been doing in neural tissue engineering, as well as in anti-adhesive space, which is kind of an accident, but really focused on trying to move what we do into the clinic so that we can help patients. So like I said, it's about the people. So Shirley Pincus is a patient who received 5 grafts, Avance grafts, because she had developed a lot of neuromas that cause a lot of pain after suffering from polio as a child. And so she was at the point where surgeons said the only thing to get rid of this pain is we have to amputate. And so she finally found somebody who was willing to use the Avance graft to cut the neuromas out, which they don't usually do. Usually they do it in an injury where you're already cut. They cut intact nerve to remove the neuromas and put the Avance graft in there. And so she's now walking and living a normal life and not having to have her leg amputated. So the Avance graft has been used in over 100,000 patients. And then the VersaWrap, this is a patient who had for tendon application, and it's been used in over 35,000 patients. So, of course, it's still about people. So these are my team members. The ones highlighted in red are some of the team members who are part of the story that I just showed you. The ones in black were slackers. No, I'm just kidding. No, just joking. The ones in black are all the other research that we're doing that I just didn't talk about today. So a lot of other cool stuff that's going on as well. And then, of course, we have funding over the years and collaborators who've helped to make this come to fruition. So we always have to remember that we have a lot of backing behind us. And so with that, thank you for listening. I'm happy to take any questions if there are any before the panel, or we can just do it at the panel. So thank you.
So we have a couple microphones for some short questions before the panel. So I'm going to ask you to use the microphone if you have questions. And don't argue with me that you can just be loud. Which wasn't supposed to scare people.
I can just call on people.
I can start.
That could be fun. No, I'm just kidding. I would never do that. Just to my students.
So does for the magnetic nanoparticle arrays where you're trying to align them, is there any attempt to do that in the body also with that, or is that solely an outside?
That's a good question. So right now we're doing it just outside the body as a to create an implant that can go into the body. But that could be very interesting to see if you could do something in the body, like especially if you have an injectable and then try to align it. And we have talked about that, but then the problem is what do we do with the nanoparticles? How do we get rid of them? Or is it going to just be rinsed and are they fine? So we've thought about it, but we haven't done any work in that space. But that's a cool possibility because we can then do in situ application and then in situ alignment if we have the magnets there. So, but.
Okay. Now we got questions.
All right, thank you. Amazing work, fascinating talk. I also had a question about the magnetic alignments, and you seem to have solved the 3D problem there that you had with the crystallization. You said that that
Yes.
didn't work in 3D.
Yes.
And so what is it that you learned from the crystallization that you applied to the magnetic alignment that allowed you to solve that 3D problem?
I
Or
think,
how did it work?
well, what we learned is we learned to start in 3D. So with the magnetic, with the in situ crystallization, we were just starting with thin films because it was easier to do the work. And then we did a ton of work and published and all this stuff and got all excited about it, and then went to 3D. And then we realized that was a limit. But that's okay too, because had we started in 3D, we never would have seen these properties in the thin film. So, I mean, it's kind of like a double-edged sword. Sometimes your mistakes are, or I wouldn't say it's a mistake, but we started one place and it still evolved into something that was completely useful in the clinic. And so we would not have been able to do what we're doing with Alafair had we not started with that. But with the magnetic templating, because we have started in 3D, so we immediately started in 3D there. So because we want to address the nerve now.
Hi, really enjoyed your talk. Not a bio person, so I apologize if this is a silly question, but
Very
also along the area of the alignment with the magnetic nanoparticles, are there applications in the body where instead of like a 1D alignment, you would want channels in multiple directions? And have you done any work with anything like that?
No, that's a great question. Yes, there are applications where we might want to do that. That's something that the Rinaldi lab, we have, oh, sorry, this is making a lot of noise, that we have continued on is how can we control pore size a little bit more tightly? How can, from a production standpoint, you want to have, be able to control it, right? But, also do we need smaller channels, bigger channels in different applications? How can we control that? And also if we have like a branch nerve, for example, there are nerves that branch. It would be nice to have one intact structure that you could have different branching applications. And then it could be applied to completely other applications outside nerve. Our interest is nerve, but it could be applied to other tissue systems. And there may be applications where you need to have, different geometries and not just a straight alignment. So those are things that we're still in the process of trying to explore. How can you do that? How can you, and that's a lot of it is on the magnetic side, which is, I'm not a magnetic expert. You gotta have good collaborators, right? Have an excellent collaborator. So yes, that's something that we're looking at is how can we fine tune this a little bit more? How can we, create variation where we might need variation? So, but good question.
Last
Especially
question.
from a non-bio person.
And we'll do the last question before we break for the changeover and the flood of students that come through at 20 after when classes get out.
Dr. Schmidt, very nice talk. Thank you. I learned a lot. And
thank you.
my question is, do we know, you may not know yet, but do you know if the nerves, if you're building a bottom-up graft and the nerve fibers, will they follow, does it have to be a bundle of conduits or can it just be a bundle of fibers? Will they follow a fiber rather than a conduit? Because you might be able to electrospray fibers or otherwise create aligned fibers that aren't hollow. So I just want.
Yes. Well, one is, yes, they will follow fibers. So if you think of the natural body's wound healing response with the fibrin cables, they're following that, those cables. That's usually with a hollow tube that you put in there. The nice thing about having something that we implant it with is it makes it a little bit easier to implant if we need an implantable, but also the outside can prevent other things from coming in. So from scar tissue from coming in. So typically the rest of the device and not just the fibers is protecting and keeping scar tissue from coming in and other things that you don't want inside so that you're just having the axon fibers regenerate. But they will follow, you could just do fibers. So there could be cases where you could, put something in there and have it follow. But just from a logistics standpoint, yeah, I'm not sure how that would work, but yes. I don't know if that answered your question or not, but yes, they'll attach to fibers. Okay, all right.
So with that,
Sorry.
so on behalf of the College of Engineering, I'd like to recognize your presentation today. And we really greatly appreciate the time you spent with us.
Well, thank you.
We're going to take a short break now while we set up for the panel discussion. So please don't go far. We'll be back in about 10 minutes, okay?
Thank you very much.